You know, the last time we had music, that's what's different. I'm like, why is it so quiet?
I know that was another thing I forgot.
Right, because we haven't done it in a while. I'm like, where is the
I
music?
know. Yeah, we're getting our training wheels back on.
I know, it's been a moment. And I could have sworn I turned off these Slack notifications and I'm still getting them. The noise, it's all right. It is what it is.
I I always have a problem with notifications. I end up just closing all of the other applications because I don't know how to silence them.
We went through that right before this, this webinar, and yeah, no, South, that didn't happen. And Bloomingdale's is showing up all like they're having a sale. Wonderful. All right.
All right, 2 minutes and then we'll get started.
Sounds good.
All right, for anyone who just came in the last minute or two, we're just going to give people another minute to come in, and we will be getting the conversation started soon.
Let me turn off my phone so my phone don't get the ringing.
Oh, that's a good idea. Okay. Okay.
Yeah, I missed the music. Next time, music. At least I get to dance around. Oh, somebody said I was glam. Thank you. Thank you, Rick. I appreciate that. I try. All right. Look at that. I see people in the building, people in the room. I'm loving this. Yes. I feel like I should have some swag around, but I don't have any with me. I tell you. I'm too busy trying to get my little lipstick together. Makeup.
Yeah, I was supposed to wear some swag today too, but
It
it
happens.
got stuck in the mail. And
Gotcha. Gotcha. I came home to a package and I was like, oh, okay. I put a little on and then I had to take it off for the other meetings I was in. All right, it is 6:05. Nava, what do you think? Think we should go
Well,
for it?
let's get started. Yep, go for it.
All right. Good evening, good evening, good evening, and welcome
hello.
everyone to another installment of the Karen's Club webinar, educational webinar series. I am your host for this evening's activities, Karen Peterson. I am the co-founder and chief patient advocate of Karen's Club, where we inspire, inform, educate, and support patients of color around clinical trials. Today's event is very special because it is Triple Negative Breast Cancer Day. I am so excited. I'm not going to introduce the guests yet because I've got a few housekeeping duties to take care of before we do that. And I know my co founder, Nava Friedman, would go, Karen, what are you doing? You haven't taken care of housekeeping yet. So I want to shout out my co founder, Nava Friedman, and tech support, Ben Fisher. On that note, there are going to be opportunities to ask questions. Please put them in the chat. Everyone will stay on mute because that's the nice thing to do, and everybody's quiet and everybody's respectful. And in addition to that, there's going to be a survey also put in the chat. And if you could feel free to fill that out and let us know how we're doing before the end of the event, I'd really appreciate that. And in addition to that, the last thing is if you are interested or know someone who's interested and needs to contact Karen's Club, please reach out to karensclub.org and a patient advisor will meet you for free, a one-on-one consultation. We can talk about all things clinical trials. So let's get started. Today, March 3rd, 2022 is Triple Negative Breast Cancer Day. And it's especially poignant to me because I am a triple negative breast cancer survivor. I am in my 5th year of metastatic triple negative survivorship. In 2017, I was diagnosed with stage 4 triple negative breast cancer, and I was given a 4% chance of survival. And I went through a lot, overcame some obstacles, and luckily for me, I got into a clinical trial and I did pretty well, and I've been, you know, doing really well since I'd say 2017. So, like again, I'm in my fifth year. I'm totally excited, and because of my good fortune, I decided to start and create Karen's Club with Nava in order to give back and make sure that other community members like myself have opportunities to talk and discuss all things clinical trial and help the community make informed decisions. So, tonight we are here with a very esteemed guest, a very talented guest. This guest has been described to me as the truth.
Oh.
Yes, you were described. I was like, really? They were like, oh, she is the truth.
What? Okay.
Dr. Melissa Davis is in the house and she is here
Thank
with Karen's
you.
Club tonight. And I am so happy and so pleased to have you with us. Thank you so much for participating in this webinar. We really appreciate you having us, being here with us tonight. And so before we begin, we just want to know a little bit about you. Who
Sure.
are you? Who is Melissa Davis?
Who am I? That is a
Yes.
great question. Let's see, it's a journey. Who am I right now? I am associate professor of cell and developmental biology at Weill Cornell Medicine. I'm also the scientific director of an international center that studies breast cancer subtypes. I'm a trained genomicist, which just means I study genetics. I have a molecular genetics PhD. And then I also have training in genomics, which means that I study all of the genes at once, right? All of the chromosomes at once. So the type of data that we generate are, these sort of large, big data sets where we can look at the interactions of all of the genes simultaneously. Um, I'm originally from the Southeast, from Southwest Georgia, actually. I'm a country girl.
Okay.
I did my PhD at University of Georgia. I did my postdoc training at Yale School of Medicine, University of Chicago. And, the rest is Black history.
Love it. Love it. Just out of curiosity, what inspired you? Like, what were some of the things that inspired you to do what you do and get into the work that you're involved with now?
Awareness. Along the way, I'll say, I made up my mind about what I wanted to do, but then when I made discoveries, not necessarily, like scientific discoveries, but just being exposed to different things, So along the way, I originally wanted to be a medical doctor. And then when I had to do my research semester at Ohio State, I had to be a scientist. It just seemed so powerful that I could make a discovery as opposed to not just, but as opposed to having a service oriented career where as a clinician, I could treat patients, which I think is very important. And that's really why I was trying to get into that. But, how much more powerful would it be to make a discovery that could change the way, diseases are treated? And breast cancer sort of entered my realm of existence during my postdoc at University of Chicago. I was really just a liaison for like the Center of Interdisciplinary Health Disparities and the Institute of Systems Biology, which is where I was originally doing my research. It's there that I met Dr. Fumio Lopate,
Mm-hmm.
who really was my first introduction to breast cancer disparities. And it was just fascinating to me what she was studying in terms of, different types of breast cancer, which, I don't think at that time in my life I really understood or appreciated that there were different types of breast cancer and that some of the worst types of breast cancer were more prevalent in people of African descent. And being a geneticist, I was like, oh, there must be something there. And yeah, I just really focused on trying to identify what those molecular markers were, what's shared amongst African Americans. I met Dr. Lisa Newman in my early career as I started out my faculty appointments. And she had already been doing a lot of work in West Africa, comparing her West African patients to East African patients.
Mm-hmm.
And again, my training, in genetics and genomics, her ability to go into the continent, and she had already, had a lot of partnerships.
Mm.
We really partnered together to try and discover, like, what is it about the African diaspora, the West African diaspora, that tends to um, promote this predisposition to triple negative breast cancer specifically. Yeah.
Yeah, I think about that often. I often think about that and think, I'm wondering what is it about our particular biological makeup. But before we get into that, so that everyone is clear about what it is, because some people might not even know. I didn't know when I was diagnosed in 2015 that there were differences. Like, what
Hmm.
is triple negative breast cancer? Just really briefly, and why is it, because of the definition, why does that make it so difficult to treat?
Right. Well, that's interesting. I mean, that's a great point. Then, it's called triple negative, meaning that it's the absence of three different markers. Those markers are hormone receptors that are normally a part of biological development, as we grow and develop, especially during puberty. For instance, it's steroid hormones that sort of dictate normal health and growth. But tumors that have these hormone receptors also tend to have a certain type of function, right? They respond to those hormones. It is those hormones that make them grow. So an estrogen receptor positive
Mm-hmm.
tumor is responding to estrogen, right? A
Right.
progesterone receptor positive tumor could be responding to progesterone. And then the third hormone receptor to give us that triple is
Mm-hmm.
human epidermal growth factor. So that's another sort of growth hormone. The triple negative tumor lacks the expression of all three of those. And that's all we really knew about those tumors, right? Is that they were triple negative.
Right.
Now, the reason why this is clinically important is because there are drugs that target the estrogen receptor and the
Right.
and the human epidermal receptor, and they're very effective.
Mm-hmm.
They're curative even if the cancers are diagnosed early. But so triple negative tumors, there is no targeted therapy because it's defined by the absence of
Right.
the targets. And so that's why it's really important to research what's actually driving the expression, or what's driving the growth of the tumors. And so yeah, so that answers your question. Don't let me
No, no, no. I'm
carry
just sitting
on.
here. I love research. I love research, but I gotta remember that it's so many other people in the room, and they're like, okay,
No, you're
we're
right.
keeping
Yeah.
up. We're keeping up. We're keeping
Definitely,
up.
definitely, definitely. Yeah.
Okay, so I'm gonna keep it simple and I'm gonna keep it conversational. I can't go back in time. I can't beat myself up, but I've always been asked this question. I get asked this question often. When a person is first diagnosed, whether it's stage 4, stage 3, stage 2, stage 1, triple negative breast cancer, what are some of the things that you would tell them? And it's different for everybody. So I'm thinking, if you were just coming across a person and they were diagnosed today with triple negative breast cancer, what are the three things that you think would be important for them to think about or to know?
I think that, I could answer this question from a couple of perspectives, and I gave it some thought, right? As a researcher, I can tell you, which is what I will tell my friends, if they got diagnosed, "Look, these are the things you need to ask your doctor. You need to know X, Y, and Z." But what I really want someone who's diagnosed with triple negative breast cancer to focus on is hope. Okay. So that's the first thing, to be positive about your life, and
Mm-hmm.
then to be open-minded about the opportunities that are going to, present themselves for your survivorship. I think that, the dynamics of the research, topography are changing
Mm-hmm.
constantly. What was not available 5 years ago has come and gone and come again, right? So, there are a lot of different opportunities for, treatment. But I think that it's important also to think about those questions that I'm telling my friends, to be armed with information. You need to think about, the staging, the information that they tell you about the tumor, the information they tell you about the options, that are quote standard of care. I think it's really important that you also have a social network, you know,
Mm-hmm.
especially I'm telling that to my friends who are, Black women,
Yes.
who do things on our own. We're very
Yes.
independent. As a
Yes.
matter of fact, it's part of our culture to not share those vulnerable details about hardships that we're enduring. But you need to connect, if it's not your spouse or your immediate circle, find Karen Scub, find people who are going through what you're going through, because it's going to be life altering, and it's
Mhm.
going to change the way you think, and it's going to change your clarity of thought, right? And so you need someone to walk that path with you, just so you can have, I don't know, different perspectives. You know what I mean?
I
Like
love that.
if you miss a detail,
Yeah.
my sister's here and she'll
Right.
remember, like I forgot to ask the doctor. I forgot to tell the doctor, that I felt bad, just to have someone walking the path with you, I think is important.
When I think about, I love that somebody in the chat said peer advice. When I
Hmm.
think about clarity, because I'm big on clarity. Clarity is important. When I think about that, I think about the word that often comes up when triple negative mention heterogeneous. And for a long
Hmm.
time, I was like, what does that mean? What does that mean? And then I didn't realize how important that word is in its
Hmm.
association with triple negative breast cancer. I can't say it enough. I can't say it enough. So, can we just talk about that for a second and just
Yeah.
get it out in the open? Like, what is that word and why is it
Listen,
so important?
heterogeneous is, let's break down what that means, right? I mean, we use it in the context of cancer, but let me give you an example of heterogeneous, right? A bag of M&Ms is heterogeneous. You're gonna get yellows, orange, what's in it? What are the colors in M&Ms now?
Green, I think.
Yeah, blue, brown,
Brown, brown.
right? Right. So the same is true of your tumor. There are different types of cells, that are growing in your tumor. And for instance, even in an ER positive tumor, it can be heterogeneous, meaning that not
Okay.
all of the tumor cells are expressing estrogen receptor. And because of that, when we treat that patient with estrogen receptor targeting therapy, only the cells that are expressing the receptor are going to be responsive. And so the same is true for triple negative breast cancer, but maybe even to a greater extent. It tends to be higher grade. And I think you're going to ask me about this
Yes,
in a
yes.
minute, right? So higher grade, what does that mean? That's part of the heterogeneity, but then the genetic makeup of the different cells in the tumor are also part of the heterogeneity. So I want you to think about cancer cells as they divide as sort of like they're rogue. You know what I mean? They're not responding to your body's normal signals of, when there's damage to some of your chromosomes, those cells are supposed to die. They're supposed to go away. But instead, your cancer cells have acquired this sort of superpower to ignore those like kill yourself,
Mhm,
signals, and they continue to divide. But because they're damaged, because the chromosomes are damaged, then every cell division is sort of a different composition of genetic material.
mhm,
And so they accumulate mutations, they accumulate tangles, in the chromosomes, and that heterogeneity sort of complicates whether or not treatments are going to work, because a lot of the time the treatment that you're given is targeting the ability of the cell to follow the instruction to die. You know what I
mhm,
mean?
mhm,
And so there's a component or a subpopulation, of cells in the mass that aren't going to respond to one type of
mhm.
therapy. And so that's why a lot of times you'll see that there are combinations of therapies, and this is
Right.
because not one, a single therapy isn't necessarily going to take care of all of the different types of tumor cells that are growing.
So, my last question associated with this. So, would we then agree or not that heterogeneous, the word, and the difference is very important to embrace as a person diagnosed with triple negative breast cancer
Yeah.
when you're thinking down the pipeline or just thinking
Yeah.
in the next 30 days about your treatment? Like,
Well,
because it's hard to sort of like, when you're given a label, you think you want to fit in there and everything. It's easy. It's an
hmm
easy fix. But to think like,
or
wait a minute, everybody's different here. Whoa, what does this
yeah,
mean? That's
yeah,
big.
yeah, that's fair. That's fair. And I think, also as a patient, one of the things you're probably going to be thinking along the way is, well, why is my doctor changing up? Like why, the first thing didn't work, does that
Hmm.
mean, something bad? Why am I taking so many different things? Why do we start out with this set of stuff and now we're doing something different? Why do I need radiation?
Right.
all of these different targeted
Mhm.
approaches are because of the heterogeneity of the tumor,
Gotcha.
right?
Gotcha.
And so be open-minded with knowing that part of the treatment is targeting some cells, another part of the treatment are
Okay.
targeting other types of cells, right? So it may seem very frustrating, it
Okay.
may seem like it's just a mix of poisons or whatever, but it's really, a strategic effort to make sure we capture or kill all of the different types of cells.
Thank you
Yeah.
for that answer. I appreciate it. Speaking of strategic, in 2007, 2021, there was a breakthrough for triple negative breast cancer. The Food and Drug Administration approved immunotherapy, Keytruda, in combination with chemotherapy before surgery, followed
Mhm.
by Keytruda alone after surgery to treat early stage triple negative breast cancer. Yes, with a high risk of reoccurrence. So those people who have been deemed diagnosed that the triple negative will come back as a high risk of reoccurrence are are more than likely pushed towards this particular regimen. Right? It's recommended. My thing is grade. Like when it says like okay, high risk of reoccurrence. I think what am I reading here? It's like the FDA guideline is again health literacy. So it's like, okay, if it's between 1 centimeter and 2 centimeters, then that's supposed to be the high risk of reoccurrence. But my thing is, well, what about the people with small tumors but with high grades of cancer? Like, I, I don't really have a lot of conversation with people about grade. It's normally about stage, it's not about
Mhm.
grade. And so, can you talk a little bit about the importance, if it's important, grade and how that factors in?
Yeah, I mean, that's a great question. And when I saw that, I started looking up some various things. I know, for instance, you're right. A lot of clinical decisions seem to be sort of focused on stage, right, rather than grade. And so let's talk about what those two things mean just a little bit so that we're all on the same page, right? So stage encompasses literally what it sounds like, like you go through different stages of a disease, right? So early stage means the tumor is very small, it's confined to a certain region, it's probably even, if it's invasive, depending on what stage it is, meaning if it's invasive, meaning, usually cancers start in the milk duct, right? So they're kind of
Yes.
encapsulated in the
Mm-hmm.
duct, but then they become invasive as they start moving out of the duct. That doesn't mean they're metastatic. It just means that they've, outgrown the,
Mm-hmm.
region that they're in.
Mm-hmm.
Um, and so staging gives the clinician an indication of how far along the tumor is. And so that also gives an indication of, how aggressive the treatment needs to be and whether they need to consider that there are cells that have moved outside of the mass that you currently see, right?
Mm-hmm.
Now, I'll also say though, triple negative tumors tend to be higher grade. So
Yes.
what are grade?
Right.
What's grade? So grade really just describes the changes that the cell has gone through in terms of whether it still looks like a breast cell, right? So in
Mm-hmm.
every type of
Mm-hmm.
cancer, wherever it originates, it was originally a part of the organ that it was in, right? It was originally a normal cell and it
Right.
looked like a breast cell. It looked like
Right.
part of the breast cell duct. As we get into higher grades, what's happening is something called de-differentiation, meaning it's losing its identity. All right.
Mm-hmm.
And
Mm-hmm.
so then as it loses its identity, it tends that's sort of that superpower I'm talking about. Like
Okay.
its identity is also a part of what it looks like. It looks that way because it's expressing those, organ specific genes and the functions that it's supposed to have. And so it's responsive to the signals that your body is giving. As it gets higher grade and it de-differentiates or it loses its identity, it's also starting to ignore those signals,
Mm.
right? It's becoming less like other cells that are in the breast.
Mm-hmm.
And a highly undifferentiated tumor tends to grow very rapidly, so
That's the key word. Okay.
yeah, yeah,
Say it again.
it grows rapidly.
Mm,
But
yes.
I also would say that a high grade tumor that's growing rapidly is also going to be larger, right? So it's going to be a higher stage. And so that's why clinicians tend to pay closer attention to stage, because stage not only defines the size, and it's going to be a larger size if it's a higher grade, but it also defines whether or not there's cells in your lymph nodes. So has it moved beyond the breast?
Mm-hmm.
If it's moved to an additional site,
Mm-hmm.
in your brain, or in your liver,
Mm-hmm.
um, so that's all a part of staging. And so clinically, if I'm a clinician and I need to think about your whole body, right,
Mm-hmm.
and what I need to do, like, is chemo going to be enough? Is
Right.
surgery going to be enough? Do we need to
Right.
institute radiation? Do I
Mm-hmm.
need you to be on, additional therapy even
Mm-hmm.
after, the first course, the first line treatment, just in case there are some cells that
Right.
escaped and I
Right.
don't see them, but they could be somewhere else growing, but they're still just too small to see, right? So that's why stage, tends to be more heavily utilized. But I also think that it's also the fact that it's de-differentiated, speaks to, resistance,
Okay.
so
Okay.
um,
We're getting somewhere now.
yeah. So, people who have high grade tumors,
Mhm.
those, I mean, how do I put this? Like the way I would, as a geneticist, define a high grade tumor
Mhm.
would be with the tools that I have, right?
Okay.
So I would define it by the genes that I see expressed. There are
Okay.
certain genes that, in a high grade tumor, we see proliferation factors,
Mhm.
but a pathologist can just look at a cell, right? Like they've been
Right.
trained to just look at the cell. They don't need to know what
And see
genes
it.
are being expressed. They can just look
Right.
at it and
And
know that
see,
it's a
like, no, this doesn't look like the original cell.
exactly,
Gotcha.
exactly,
Gotcha.
exactly. And so, but those things are correlated, right? So the tests that are done to determine if a patient is high risk of recurrence, for instance, incorporate the same genes that are expressed in a high grade tumor. So what am I saying to you, Karen? I'm saying that grade is actually a big part of it, even if it's not called such,
Okay.
from a clinical standpoint. You know
I
what I
think that's
mean?
a
It's
really important information for people to hear because I didn't understand that and didn't know it. And so, again, I can't go back to Karen in 2015, but Karen Peterson now can pay it forward. So I really
exactly.
appreciate that. I
Yeah.
appreciate you identifying that. There's one other thing I wanted to talk about when we talk about clarity and we talk about identifying things and we talk about treatment options if things have escaped. How do you sort of like identify, like, what are the sort of the tools when you talk about tools, like the biomarkers, like a person coming in who doesn't know, who's just been diagnosed and who's thinking, well, I've heard her talk about genomics or I've heard her talk about genetics. Why are those tools useful and what are those tools? Can you speak briefly about that?
Briefly. Okay, I'll try to be brief.
I'm sorry.
No,
I'm
it's all
sorry.
good. It's all good. No, I just know I'm long-winded and now you're talking about my specialty. So okay. Let's see. How do I want to put this? So, I mean, there are different types of genetic tests, right? There's a genetic test and then there's a genomic test.
Right.
Okay. So, a genetic test is when I'm looking at a certain gene, like BRCA, right? And I want to know whether or not your tumor or even in your germline, even the DNA, and by germline, I just
Hmm.
mean like your chromosomes that you've inherited from your parents,
Right.
right?
You were born with.
Exactly. Whether or not they have a certain mutation, certain types of mutations in key genes that we know can drive the malignancy of a tumor. So, for
Hmm.
instance, BRCA's function
Hmm.
is DNA repair.
Right.
If you get a mutation or were born with a mutation that takes away the function of BRCA, then your cells are prone to accumulate more mutations, and that's why you're at a higher risk of cancer because the accumulation of mutations leads to malignancy, right?
Gotcha. Gotcha.
So a genetic test is going to be given to a patient who has a family history
Hmm.
of breast cancer because we want to know as early as possible, whether or not you are carrying a loss of function mutation in BRCA, for
Hmm.
instance. But it's not just BRCA now. Like there are dozens of genes, who have similar function.
Like a knot.
Um,
Like a notch,
exactly.
right?
Yeah, yeah. Notch is one, Notch P53,
Mm-hmm.
Paul B,
Mm-hmm.
Rad 51, like there's a lot of genes now, which is why it moved from a single gene test to what we call a panel, right? So this is
Gotcha.
a mutation panel, right? It's a flight, or a lot of
Mm-hmm.
genes together.
Mm-hmm.
A genomic test is more about the structure of your chromosomes. And it's sort of an indirect way of knowing whether or not a mutation that we may find or may not even find, like we only know a handful to look for, on the mutation panels. But I can look at your whole genome and see a pattern of mutations that would indicate to me that there's an issue with DNA repair, or there's an issue with the way your cells are supposed to resolve your chromosomes when they divide. Like I can see the imprint of that, right? And how else would I describe it? Like, not knowing the cause of, a scrape in a paint, you know what I mean, on a car or whatever. I may not know what the cause of it was, but I can see that there's damage here. I see the damage and I know that it's not getting fixed, even if I don't know why.
Right.
I know that the damage is there. And so, and I might look at BRCA and I might find like there's no mutations in BRCA, right? So that's why it's important to have both,
Okay.
because there are no mutations in BRCA, there are no mutations in any of these other genes, or more
Mm-hmm.
importantly, depending on your ancestry, this
Mm.
gets even deeper, right? But
Okay.
depending on your ancestry, there may be a mutation in some of these genes, but they haven't been researched. And
Gotcha.
so when they come back on your gene panel, as we found a mutation in BRCA, but it's a variant
Mm-hmm.
or
I've
a
seen that in the
change of
report.
unknown
Right. I've
unknown
seen that.
significance, right? You're going to see that a lot in people who are of non-European ancestry,
Mm.
because most of the knowledge that we have about BRCA mutations was generated in people who were of European descent,
Right.
right? So, but, fear not. The
Right.
genomicist is going to come and look for like the patterns of the structure across all of your chromosomes. So even if this variant of unknown significance is something that your doctor may feel like he can't say anything about because it's unknown or uncertain, well, that's okay. We see, the landscape is scattered with those types of mutations that we anticipate seeing in someone who's carrying a loss of function mutation. Now, you as a patient, you asked me, when should you get those types of tests done?
Right.
I'm
I
gonna
think
be
that's an important
honest,
conversation.
like,
Okay.
it's not up to you. Not directly. It's up to you in the sense that where you get your care and whether or not they have the capacity to do that type of testing, and whether or not your team of clinicians deems it to be useful, right? So, for instance, that whole genome test, it's, there are a few places in the country where you could get, what we call CLIA approved, meaning that, it's a lab test that the FDA will allow you to use for
Mm-hmm.
diagnostics, right? So there are a few places that you can get such a test done, but that's not what you're going to typically find at like your community serving hospitals,
Gotcha.
unless they're partnered with academic centers, right? So
Mm-hmm.
that's sort of that's sort of like the inside information that you need to have access to, a place that has the capacity to, or access to a place, that has the capacity to do the whole genome sequencing, types of testing. But that's still kind of not first line. That's just to give the doctors more information, right? First line genetic testing is still, by and large, just whether or not you're harboring a
Right.
mutation in a certain set of genes. And
Gotcha.
that's something that's typically done no matter where you are. Current clinical guidelines are that any patient with breast cancer should get genetic testing.
Mm-hmm.
Yeah.
Okay. There's a lot I could say about that, but I'm just gonna leave it there.
Okay. We're gonna
Um,
we can talk about it.
I,
Me
um,
too.
um, I guess what I think when I think about what you just said, what stuck with me most was where you get your care. So the importance of that. So I feel like we should talk about that for a second. Like,
Sure, sure. So, I'm not here to give a commercial,
right.
for New York
No, but do your
Presbyterian.
thing.
Well,
Do your
I'm not,
thing.
I'm not gonna say that. I'm not. But I am gonna say is, and I just saw this pop up in the chat. Yes, you can advocate for yourself. Listen, if you go to a doctor and they, tell you what they're going to do, but you've already armed yourself with information and you didn't hear them say something that you thought they should say, and then you ask a very direct question, well, are you going to do X, Y, and Z type of test? That is a light in their head that will say, okay, maybe I should consider this, or at the very least, I should explain to the patient why it is or is not appropriate, right? And so I think just bringing the doctor to the point that you let them know you're aware of all of the different types of diagnostic options that are available will sort of compel them to tell you yay or nay why they chose to do it or why they chose not to do it. um, and then if you're not comfortable with their explanation of, like say for instance, you get diagnosed and they tell you, oh, it's not worth doing any type of, genetic test. We're just going to do X, Y,
Right.
and Z.
Right.
You can insist on it. You can ask them why they wouldn't give you the genetic test, and, even get a second opinion, you know.
Okay. I think it's really important. Again, I've had a lot of opinions from people saying, well, it's about where you get treated at. If you go to an NCI designated center, but oftentimes I think sometimes we do get lucky and we do have those clinicians in community based
Yeah.
um, that do their job and are
Yeah.
interested in making sure that they give the best quality care that, with the tools that they have. So,
Exactly. And that's what I mean by, or they have access to it, right? I
right,
mean, even if there's not a sequencing center on site, or,
right,
your cancer center isn't NCI designated, um, all doctors have access to information, you know what I mean? And all doctors have the ability to order certain types of tests. So even if it's not something that's offered directly in their healthcare system, they can certainly, identify a place to order the test from.
okay. I'm
Yeah.
going to ask one last question before I start digging into these questions from our lovely Karen's Club members and everyone who's attended the webinar. The question was asked the other day when I was part of a panel, and a nutritionist was asked the question and she gave an answer. And I just want to get it out here and finally deal with this. Does sugar feed cancer? I'm just curious. Please, please answer the question.
She said, please answer the question. Listen, sugar feeds all cells,
Okay,
right?
okay,
And we already know that tumors, cancer cells already have like the superpower to ignore the signal that says stop, right? And so how much more are they going to proliferate if you're feeding them more of the basic cellular structure, the basic cellular energy that they need, to proliferate more. Um, that said, any any type of diet, a healthy diet is a healthy diet. You know what I mean? I'm not saying that, and I'm not an expert, I'm not an expert nutritionist. There's, new fields of nutrition oncology,
right.
coming forward. There are a lot of clinical trials even that I was on a plenary with AACR, last year. One of my colleagues spoke about a tailored diet that matches your therapy.
Mm,
It's not always low sugar. It's not always low fat. It really depends on your metabolic makeup and the type of therapy that you're on. So it's not as simple as saying don't eat sugar. You may need to eat more fats. You know what I mean? Like that's where a team comes into play, having a team of clinicians who are not just the surgeon and the oncologist, but also a nutritionist, or, an immuno-oncologist, or a
yeah.
cardio-oncologist. All of these people come together, um, in some instances, in something called a multidisciplinary conference, right? Where
Mm-hmm.
they talk about many different aspects of a patient's, makeup, and, what's beneficial. So does sugar feed tumors? The evidence in mice is undeniable that, high sugar diets in mice make the tumors larger. And we do know that people who are obese, and of course eating a lot of sugar will lead to obesity, do have, a higher risk of cancer straight up. at baseline, but
Mm-hmm.
then also have a higher risk of recurrence.
Okay.
So
All right. You've
whether
answered.
it's direct or indirect,
That was fair. Nava, the co-founder of Karen's Club. I know we have a lot of questions. Do you want to jump in and start popping those questions out?
Yes, we had a
Wonderful
very active,
conversation.
sorry, we had a very active chat. So hoping we can get through as many as we can. And I tried to do some consolidation. But we had a few questions about recurrence. So does triple negative breast cancer have a different or higher recurrence than other breast cancers? And how should we be surveilling for recurrence? How often and using what methods?
That's a good question, doctor.
Okay. Um, I'm not an MD, okay, so I'm going to give you my scientific viewpoint, but I will tell you that recurrence is basically dependent on what is left over after the first line therapy, right? So recurrence is not the same as a secondary primary. I'm using some jargon, so let me break it down, right? So you have a tumor, you get treated for the tumor, and then at the end of whatever the treatment cycle was, whatever those, different drugs or treatments were, then there is an assessment of what is left. And usually that's surgical intervention, right? So after the tumor area is resected by the surgeon, the pathologist takes a look at it and says whether there is residual tumor and what's left, right? The more that's left, the more likely you will have a recurrence. And a recurrence means that the tumor comes back, meaning it went away for the most part, but then it starts growing again, right? So that's a recurrence when the mass becomes palpable again and you start to see the tumor come back, right? As opposed to, and I just want to clarify this because I don't know if everybody is aware, but a second primary, excuse me, is when you've been cured of therapy, there was no residual, we didn't see anything left, you're in complete remission, and then 5 years later, you get cancer again. That's a second primary, right? So the cancer came back.
Mm.
So a recurrence score is a combination of the aspects of what the tumor looked like when it was first diagnosed. That's where those genes come in, the genomic signatures, all of the different types of genes that are proliferative signals, the grade of the tumor, right? All of those come into play. And then like there's a special algorithm, right, that they put together. Is it a BRCA carrier patient? Did it have hormone receptor, X, Y, or Z? And I think I'm missing the question, but I think, as far as triple negative specifically having a higher recurrence, that gets back into the heterogeneity.
Mm-hmm.
It depends on the composition of the tumor. And I'm going to be honest with you, like statistically, yes, there probably is a higher recurrence of triple negative because it's harder to treat. It's higher grade. It's probably going to come at a later stage, which means it's probably going to have some node positivity, and surgeons do their best to try and remove all of the nodes that could possibly have tumor. But it only takes one cell left behind for a recurrence, right? And they don't have the tools currently, not all of them. There's some space age Star Trek type of technology, out here, but not all of them have the capacity to see the cells as they're doing surgeries, right? And I think that that's part of the research, right? That's part of what we're doing in terms of trying to really identify better ways of treating triple negative so that for instance, estrogen receptor positive tumor cells that escape the region, we can still treat those even if we can't see them because the drugs are targeting them no matter where they are in your body.
Mm-hmm.
But for triple negative, there's no like targetable thing.
Right.
Now, I'll also put an asterisk and say the chemo now is starting to be loaded on antibodies, and we call these conjugated antibody therapies.
Mm.
And those antibodies are supposed to be able to identify the tumor cells, and basically it's like a payload, right? So it's like a truck,
Mm.
a delivery truck. So the chemo is loaded onto the antibody, and then the antibody finds the tumor cells. And so that type of sort of conjugated chemotherapy, antibody chemotherapy, is more promising. And I think there are clinical trials that indicate patients who have a high recurrence score are eligible for those types of therapies.
Conjugated. I'm writing that down. Very interesting. You know, I'm going to research that tonight. Okay, thank you. I learned something today.
All right. Yeah. So did I answer your question? Did I answer the question? Was there a part I left out?
I think it sounded good to
Okay.
me.
Okay.
But well, I'm sure we'll hear from the audience in the chat if there's follow-up.
Okay. Yeah.
While we wait for that, question just on definitions. After treatment ends, is remission the same as no evidence of disease? And if not, what's the difference?
So remission is when, yes, there's no evidence of disease, or it can also be called pathologic complete
Response.
response. Yeah.
Yeah.
So we can't detect any of the tumors anymore. Know where we saw them before, even if you had a scan, like if you had stage 4, there's no detectable cancer. So you're in remission.
Okay, so it's sort of the same thing.
Yeah.
Oh, okay. All right, yay. I never use that word. I just use complete response. But yeah, okay,
Yeah.
good.
Remission means, in remission for some, could also potentially mean that they're no longer even taking therapies, right? You're in remission.
Oh,
So yeah, the
oh,
first diagnosis is over. You're all good. And hopefully you'll stay that way.
thank you.
We had a few questions also about genetic testing. So a few folks who had been diagnosed 2 years ago, some 4 years ago.
Okay.
Should they do genetic testing if they didn't do it originally, or if they did it a few years ago, is there any reason to do it again?
That is a great question.
Mm, I
Um,
like that question.
yeah, I do too. I like it for a couple of reasons. Well, I'm a geneticist, so I'm always gonna say yes, do the
Yeah,
genetic test.
yes.
But I think, from a clinical standpoint, what's also really interesting, like, okay, we talked about recurrence.
Mm-hmm,
So if you had a genetic test on your first tumor, a genomic test on your first tumor,
mm-hmm,
then there's some signals that we're going to see in the genetic signature that point to whether or not a certain type of alternative therapy or clinical trial might, be beneficial to you. And so after first line, if you blow through first line, they'll have that data to then go and identify additional drug targets that may not, about to kick it over to you, Karen, may not have been typically the drug of choice for the type of cancer that you have. Right. So, for instance, some lung cancers may have HER2 amplifications, although the HER2 amplification was something that was typically a breast specific diagnosis. And so then the drugs that were developed for that HER2 amplification could be working well for someone with lung cancer, but we wouldn't have known that if we didn't do the genomic test to identify that this lung cancer had a type of genetic alteration that you would normally or typically commonly, find in breast cancer. Right. So yes, get the test. But then if there's a recurrence, and this comes back to heterogeneity, sometimes what's growing isn't the same as what was growing before.
mm-hmm.
And so you need an update. You need an update on everything,
Yes.
whether it's a hormone receptor status. Like, there are a lot of times when women who had estrogen receptor positive tumors will recur with a triple negative tumor.
Huh?
So yeah, I mean, testing again is very important.
I think
I
testing is important. It opens up options. The testing that opened up the option for me was genomic testing. And again, a lot of people were surprised by the fact that the clinical trial that saved my life was not just specifically targeting triple negative breast cancer and mutation was going on. It was a clinical trial that was open to sarcomas. And as long as you had something solid, they were going to, put you in the trial. And it was open to melanoma, small lung cell, urethra, and they opened the door. We were the last ones that they invited into the trial. And the reason why the trial worked for me was because I had a lot of TILs, tumor infiltrating lymphocytes, these spider cells, but they just happened to be on a coffee break. So the particular drug that I was on, the IL-2, sort of like got these spider cells together, and then the PD-1 inhibitor that was the combo with it sort of like took back the cloak and sort of like help the fighter cells do their job better and kill the cancer,
I love
recognize the
that.
cancer and kill the cancer.
I
So
love that explanation. Yes. That's amazing. But I want to also just follow up too. So what I was just describing was genetic and genomic testing on the tumor, but germline testing, just to know your, your not your receptor status, but your cancer gene status, could also be very important as well. Just to be aware of your risk and your family's risk. But yeah, I like that coffee break. We use the term exhausted T cells or exhausted TILs, but I like the coffee break one because that gives me hope. Like a cell that's exhausted, I can't do much with. But a cell on a coffee break, like, okay, break's over. Like get back
exactly.
to work, get back to work. I like that. I like
That's
that.
what they did. That's what they did. So I appreciate that. Oh, thank you, doctor. Okay, what other questions?
Um, all right. So I think we have time for one more. I do want to know we're not going to get to everything, and there were some specific case questions. So Karen, maybe we can figure out how we can follow
Yeah.
up on those. A one-to-one is a great way to ensure that we can connect and either help you figure out how to get it from your doctor or maybe forward some things over to Dr. Davis. But last question for this, because it connects to a topic we've been talking a lot about, is when you talk about recurrence scores, how is a high recurrence score actually determined?
Hmm.
Hey, let's get into some math. No, I don't, so there's a caveat to the current so there are tests. I don't know if I can use a commercial name, but I'll just say like there's a set of genes whereby if we look at the level of expression, either up or down of a combination of genes, that again gets into the biology of the cells and it tells us, how aggressive they are, how proliferative they are. This in combination with, for instance, the staging. Recent studies have actually shown, though, that recurrence score doesn't necessarily operate the same for all patients.
Hmm.
Um, but how is it calculated? So it's literally like math, like there's an algorithm where, let's say there are 20 genes, out of the 20 genes, if the top 5 have high expression, the next 5 have low expression, and then you see just the presence of altered networks, gene networks, each one of those sort of is a point. You know what I mean? And so then you additively decide when you get to the end of all 20 genes, what is the score? And then there's a threshold. So if the score is like taking a test, if it's the score from 1 to 100, anything above 40 is considered high recurrence, but something below 40 is considered low recurrence. Well, it turns out, this sort of threshold cutoff thing, sometimes, it's not accurate. You can have a cutoff of a lower number and still recur. And so that's what I mean when I said it's not performing the same in all patients, that a lower threshold in certain patients is still a high score. You know what I mean? But I think part of the complexity with the recurrence score, and I mean, there's been a lot of research around why they use the cutoff that they did, you know what I mean? But this gets back to heterogeneity of the tumor, but it also gets back to diversity in the clinical trials. The initial clinical trials where they set the threshold of what a high score is versus a low score was not very diverse. And so
Hmm.
now when they try to implement the same threshold across the board for everyone, it's not really holding up, as well as they would like it to. And
Hmm.
so now they're going back, we're going back, we're doing it again, looking at, what is the better threshold for, certain patients. But then, here's my job. Why? Like, why is the threshold different in these patients? Because there are more genes to be considered. So the current test only looks at 20. Maybe there's one more, that's not included in the 20. Maybe we need 25.
Yeah.
Maybe it's a different set of 20. right? And so, it's still a work in progress. But by and large, it tends to be, if it's high, it's high, right? Like no matter who you are, if you have a recurrence score of 90, that's a high risk of recurrence. The issue is sort of when you get to where the cutoff is, around 30s and 40s or even 20s, whether or not that's indicative of high risk.
Okay, we're getting close to closing time. I just want to give you the last word because I think it's really important. Um, how important do you think clinical research is in triple negative breast cancer as we go down the line? Like, how important? Let everybody know how important that is.
It's imperative. I mean, there's no way we're ever going to make standard of care better if we're not doing research. And we have to do clinical trial research. We can do research in mice and in cells and petri dishes, all day. But until we can actually test these, treatments and observe in tumors of various ethnic groups, various subtypes, like right now, I think there's, depending on which tool you use, there's either 3 or 6 different
Yes,
subtypes of
subtypes.
triple negative,
Exactly,
right?
exactly.
And so we need more information about each one of them and to drill down into like, well, what's the real driver of the malignancy in each one of these, different subtypes. And we can only do that with more research, more patients, enrolling in research. And I'm not talking about even clinical trials at that stage. I'm talking about just give us permission to investigate what the genomic and gene expression profiles are in your tumor so that we can better inform the knowledge base, right, about the diversity, so that then when somebody comes to the table with a drug, we can tell them, well, you're only going to treat, 5% of the population, and we need somebody to really focus on this 10% because there's nothing currently, focused on that, theoretical, 10%. So yeah, it's imperative. We need you guys. Definitely. We need you guys.
This has been such an impactful and just inspirational conversation. I feel like I learned a lot. I feel like I could go on and on, but I'm not going to monopolize your time because I know you're very busy. I just want to thank you on behalf of Karen's Club and everybody who
Absolutely.
participated in this. You are wonderful. You are a breath of fresh air and a wealth of
So are
knowledge.
you.
And we truly appreciate you for doing this. We really do.
Thank you. And yes, you guys can definitely have my contact information. I don't know if you guys already, sent this out by invitation, but thank you for having me. It's always a pleasure, to have these dialogues. I learn too. I learn, especially when I'm failing to make my point, and I need to explain something better because the jargon, someone asked about literacy, health literacy,
Yes.
it's not just on patients to learn, it's on us to teach, and I think we need to do a better job of teaching. So always happy to do stuff like this. Thank you for inviting me.
Thank you so much. Thank you to my co-founder Nava. Thank you to Ben for helping out with tech support. Doctor, once again, Dr. Melissa Davis, you are the truth. Thank you everyone for participating in another educational installment of the educational webinar series at Karen's Club. We truly appreciate you guys for coming. Feel free if you need to contact us, go to karensclub.org and we're here. We're to support you and talk to you. Thank you so much. We appreciate you. Thank you so much.
Thank you. Have a good night.
Good night, everybody. Thank you for coming. This was great. Thank you.
Absolutely.